Now let's tackle the question of how a memory is stored. Well, for a long time, scientists suspected that memories, learning and memory, might have something to do with the junction, the synapse between neurons. This idea goes back a long way to some of the early neuroanatomists like Ramon y Cajal. He was an expert neuroanatomist. Here he's sitting with his old microscope there, and he drew fantastic pictures of what he saw under the microscope. And here we see a neuron, its cell body, and the dendrites, and he noticed all the little spines on the dendrites, and he drew them. And he hypothesized that the bumps on the dendrites were connection points with other neurons where changes associated with learning might occur. So this idea that learning and memory might have something to do with the synapse goes back a long way. Today, there's much evidence that Cajal was correct. Learning and memory are rooted in changes at the synapse, including the growth of larger spines where the axon terminals connect to a dendrite. In the 1970s, a fundamental discovery was made about the biological basis of learning and memory, and it occurred in the hippocampus. Now, this was after HM, so people already knew that the hippocampus was a critical structure for memory processing. Recall, it's underneath the temporal lobe. Here's a cross-section. There's the hippocampus, and here we see it. The cells are stained. Here's a magnification, and again, the little blue-purple bits here would be the cell bodies of hippocampal neurons. Now, in the 70s, scientists developed the ability to keep thin slices of hippocampal tissue alive for hours. They bathed it in a nourishing solution, and then they were able to insert stimulating and recording electrodes into separate hippocampal neurons and then influence how they talk to each other. They could study the electrical and chemical properties of live neurons. And here's the basic discovery. We'll simplify it here with this diagram. We have two hippocampal neurons here. We'll call the one on the left the input neuron and the one on the right the target neuron, or the presynaptic cell and the postsynaptic cell. Well, what they did was they electrically stimulated the input cell. They generated one electrical signal, and that caused some small amount of chemicals to be released, and consequently a small electrical response in the target cell. It did not reach threshold, so it did not generate its own output signal. Then what they did for that same neuron, they simply zapped it. They gave it a high frequency electrical stimulation. So we have lots of electrical signals per second racing down to the terminal. Much more neurotransmitters, much more chemicals are released. So they generate a larger electrical signal in the target cell, and that cell passes threshold, so it generates its own electrical output signal called an action potential, AP for action potential. And now here's where the fundamental discovery was made. They're going to now, after giving high frequency stimulation to this same input neuron, they're going to go back and just give it one test pulse. They generate one electrical signal. And what they found was an increased response in the target cell. It's as though the high frequency stimulation somehow caused the synapse to get stronger in a sense. The communication was more effective and more efficient here. This was called long-term potentiation because this increase in response lasted for hours. Now, without going into a lot of detail, here's just a brief summary of some of the things that have been since discovered about this increased synaptic efficiency. Let's say here's a baseline communication. We got the terminal. We have a spine here. We have neurotransmitters in the terminal. When they're released, they open up channels on the spine, and these channels allow charged ions, charged particles, sodium ions, into the spine there, and that's the electrical signal. What they found was this LTP seemed to be associated with an increased number of receptors. So for a given amount of chemicals, you're going to have a larger electrical signal. And then over longer periods of time, the spine itself enlarged. So you get physical growth of the spine. And that enabled the spine here to have a more permanent set of an increased number of receptors. So this change at the synapse, this increase in synaptic communication here, would be the biological basis of learning, and if that change could be sustained, the biological basis of memory. Now, scientists also noticed something interesting about LTP. Synapses that actively take part in exciting the target cell get strengthened, but other synapses that are not currently involved in activating the target cell were not strengthened. We're going to call this synapse allocation. So just as we said that certain neurons in the hippocampus might be allocated to forming a certain memory trace, so too at the level of synapses. Certain synapses can be strengthened while others are not. Only those synapses that are currently active and take part in firing the target cell, only those synapses will be strengthened. Other synapses will not be strengthened. Now, in the middle of the last century, a psychologist by the name of Donald Hebb made a prediction about this type of synaptic change. He said when an axon of cell A is near enough to excite cell B, or repeatedly or persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells such that A's efficiency as one of the cells firing B is increased. Now, that's a mouthful, so we'll summarize it as cells that fire together wire together. Now, we're going to call this synaptic plasticity, and that refers to the capacity of the synapses to change their strength. During learning, a lasting increase in synaptic communication is called long-term potentiation, or LTP. And again, we're going to have a little icon. So here is the synapse before learning, and notice it's a small little terminal there, and the icon for LTP will just be a bigger terminal, indicating that this synapse is now a stronger connection. And that happened as a result of the learning episode. Now, the original discoveries were made in the hippocampus, but since then, LTP has been discovered in many regions in the brain. So synaptic plasticity occurs in many regions and involves a biological growth process that can be maintained for years. Thus, memories are stored as changes in synapse strength between the brain cells that were involved in the learning episode. Now, what is it? What is this growth that's occurring? Well, brain cells are living machines that can build new cellular parts called proteins in response to learning events. So here we have, again, a brain cell. Here's the nucleus of the cell, and there's DNA here. And we'll be learning, or perhaps you already know, the DNA is the recipe to build proteins. And the idea is when a brain cell is involved in a learning event, it's going to start to build some materials. Those materials, you can think of them as sort of like bricks and wood and nails and stuff. Those materials will be sent to the relevant synapses that were involved in the learning to make them grow, to strengthen the connections at those locations. So what we have then is that learning is causing a physical growth at the synapses that were involved in the learning. The spines are getting physically larger. There's more receptors inserted into the membrane. Perhaps more chemicals are released for each electrical signal. So we have a strengthening of the connection as a result of learning. And what this means that for any particular signal that comes down this axon to the terminal here, it's going to have a larger effect on the target cell as a result of this growth process. So synapse growth can be sustained for long periods of time, thus making memory retrievable over long periods of time. So we want to think about long-term memories as involving structural changes at the synapses involved in the learning. If we return to our picture of the hippocampus, again, this picture showing the allocation of hippocampal cells to a memory trace for a given event. Now let's look at it from the point of view of synapses. And again, this is obviously a simplification here, but now we have a bunch of synapses in the hippocampus. Prior to learning, these synapses have a baseline strength. But after learning, a certain subset of those synapses will be strengthened. So the green cells would be the allocated neurons, and those neurons that were activated by the learning episode, they will enjoy the LTP. So neurons that fire together wire together. Notice in this case down here, only this pathway has a strengthened synapse, whereas this neuron did not get activated by the learning event, so its synapse onto that target cell is not strengthened. So some hippocampal synapses will be strengthened, that's LTP, to create the memory of the event. Different learning events activate different sets of neurons. So the hippocampus stands ready to make different kinds of memory traces for different learning events. But for this particular learning event, that set of green cells was allocated to make the memory, and the memory consists of changes in the strengths of the synapses between those allocated neurons. So Synaptic plasticity. The strength of neuron communication can change. And this is what we mean by synaptic plasticity. We've identified LTP, long-term potentiation. That's when the synapse gets stronger. But there is the corresponding phenomenon, LTD, long-term depression, weakening synapses. Changes at the synapse involve biological processes, so proteins are produced, cells are making proteins to physically change the size and shape of dendrites and release more chemicals, etc. at the synapses. And synaptic plasticity accounts for learning and memory. Learning causes the plasticity, and the maintenance of that plasticity accounts for memory. And we'll wrap up this lesson by considering, why does the brain undergo the system consolidation? One way to look at it is the hippocampus has relatively fewer synapses compared to the cortex, and this means that newly encoded memories might interfere with older memories within the hippocampus, and system consolidation solves this problem. So if we think of this hippocampus, it has a finite number of cells, therefore a finite number of synapses, and there are just many more neurons in the cortex, tens of billions of neurons, each with thousands of synapses. So there's just a lot more capacity to store information in the cerebral cortex compared to the hippocampus. So system consolidation then is transforming a memory from a more limited capacity, fast-acting memory system to a larger capacity memory system in the cortex.